Are there any announcements that need to be made? Okay, let's get going then. So it is a great pleasure to introduce Dr. Anna Simush. I'm sorry about the pronunciation of the last
It's
name.
okay.
I'm trying. Anna is from Portugal. She did her PhD work at the Natural History Museum in London, as what that was in collaboration with Kew Gardens as well as the Singapore Botanical Gardens. She is a tropical plant biologist and is really 100% a taxonomist. Is that right, Anna?
That's absolutely correct.
Absolutely amazing. She's done two different postdocs. One was located in Singapore, continuation of work at the Singapore Botanical Gardens, and then a second one in Brazil. And then she also took a teaching position in Brazil for a little while as well. She's finally ended up back in Portugal in her home country, and she picked up a fantastic job working at the Kew Botanical Gardens. For those of you who are not botanically minded, this is one of the flagship botanical gardens of the world. It is a phenomenal place, and I am very envious of the job that she's been able to obtain. She's going to talk to us about convolvulaceae. I think I put together a title accidentally that talked about conservation and biodiversity. I apologize for that, but
It's okay. We've all done that.
taxonomist, and so she's going to talk to us about a lot of the work that she's been doing over the last few years. And I'll give way to you, Anna, and let you take control. Thank you very much for being here today. I appreciate it.
Well, thank you, Matt, and thank you for the lovely introduction. I'm really glad to be here today. Thank you for the opportunity. For me, it's always great fun to meet new people. I'm European. I've worked in the tropics. I don't always extend a network and get to meet students from the US, so this is really good for me as well. It's always great fun. I hope I'll be able to give you a bit of entertainment today. What I've put together, just so you understand, because I wasn't sure what everyone's interests were, I've put together bits and bobs, as we say, bits and pieces of different presentations I've had. So a bit about me, a bit about Convolvulaceae, and a bit about the institutions where I worked. I hope it's not too long. If you think it's getting too long, you can just kind of tell me. But basically, I didn't get too technical. So if someone at some point would like to know more of the details of my work, I can eventually either invite you to one of our talks where we will talk a bit more about systematics and taxonomy, or I can give a talk just about, the work that I do in detail. But for today, I thought, let's make it a bit lighter and just talk about me. And sorry, can I just
But don't be sorry. You go do
no,
what you
no, no.
do. It's okay. But family lives overlap with academic lives sometimes. It's fine. No one
Anyway,
this about it.
I'll get started and
While we enjoy a momentary break, are there any other announcements we'd like to talk about?
sorry.
Don't be sorry. It's fine.
I think it's sorted, but I'm not sure. Give it a try. I need to share the screen, so that should be okay, right? I should be able to share the screen. Let's see how that goes.
Lynn can give you host control if not.
Okay, so if that works, there we go. Share.
Nice.
Is that okay? Do you see
Yep,
that
we
the
see it.
presentation? Okay,
Mhm.
and you see the presentation as in the actual slides, and probably I'll be seeing the notes, right? You're not seeing my notes.
We don't see notes. We just see.
Good. Okay, so this is a completely different title from what Matt suggested. I might switch off my camera as well. Stop video. Yeah, there you go. So this is a bit of a different title from what Matt suggested, but this is a talk I've been giving to people when I introduce Convolvulaceae for those who have never heard of it. So from sweet potato to LSD. Put your seatbelts on, right? So, so So I'm a taxonomist at the Royal Botanic Gardens Kew. I work at the Africa Identification and Naming team. Let me just, how do I go about this way? So I've joined the Africa Identification and Naming team last June. So it's quite recent, as Matt has said. My skills and research interests are primarily related to taxonomy, molecular systematics, and conservation. And I also have a keen interest in palynology. At Q, I'm also involved in some teaching. It's something I've done as well before. So I do participate in the teaching in the MSc course. And in addition, as you've seen, I'm the mother of a 5-year-old little human who assists with my work. So general introduction of me. So this is my contact details. You'll see this slide again at the end, so don't worry if you don't need to write anything down. But if you want to get in touch, you'll just see this again. My main research line is taxonomy and systematics. So it means I produce floristic accounts, taxonomic revisions, monographs. I do field work in the tropics, collect specimen samples, and we just identify and name actual specimens, not just of Convolvulaceae. But of course, my main focus is this family, Convolvulaceae, and Africa. That's what my job is about. So Convolvulaceae, this family, what if you haven't heard of it, so we get a bit technical. Following APG, so it belongs in the Solanales order and is sister to Solanaceae. So you will know Solanaceae because it is a family of potato, tomatoes, peppers, eggplant, or aubergine, or how you would call it. I may get mixed up between British and American English, and I go back and forth because it's not my native language, so you'll excuse me. And that's Convolvulaceae, so it's sister to Solanaceae. So a lot of people ask if sweet potato is the same family of potato. It's not, but actually they are sister families, so they're not that distantly related. Okay, so you will easily recognize this in the field with these campanulate corollas, these flowers with this particular shape, with the bright colors. You can't miss them. You might have seen them many times, even in the US, because there are quite a few species in temperate regions. Although having said that, they are widespread and they're much more diverse in the tropics. This map is from JBIF, so you get distribution points. The reason you get a lot in the temperate region is because they're much more collected in the temperate regions, but in terms of diversity, they're much more diverse in the tropics. It's just seriously under-collected. So you see that gap there in Central Africa. That's just because we don't have enough distribution points, enough collections, but diversity is expected to be really high there. So it's widely distributed, and also ecologically it has a really broad range. So this is from a presentation from Brazil, so of course it refers to habitats in Brazil, but you'll find them in very dry regions, from cerrado, so like savanna-like habitats, to desert, ocean shores, primary forests, just basically anything you can think of. But finally, they do have a preference for disturbed habitats, so you will find them very often in open areas, near cultivations, roadsides. It doesn't mean that they're being invasive or they're not native. So this is something people sometimes are not aware of. It's just that they really like these open areas. And sometimes they're native, they're endemic, they're endangered, but they just occur on the roadside because that's what they like. Okay, so okay, so of course you will have known many of these Convolvulaceae. They're bindweeds, so Convolvulus, morning glories, which is more from the genus Ipomea. They are widely cultivated, they're really important in horticulture. And actually in Japan, for those who don't know, there's a dedicated festival to them every year, a 3-year festival where people trade seedlings, there are competitions, there's all sorts of merchandise with morning glories. It's a really big party. So anyway, it's just a fun fact about them. And most of all, they are important for the crops. So there are two main food plants in Convolvulaceae. There's sweet potato, it's Ipomea batatas, and water spinach, Ipomea aquatica, which is really common in Asia. So in the Americas, people usually don't know or haven't heard of this water spinach, but in Asia, they have all sorts of names in Indonesian and in Thai and in Chinese. And if you go to any restaurant and you ask any Asian restaurant, if you ask for stir-fried morning glory, they will know what it is, they will have it. It's really, really common. So these two plants together, either in the Americas, Africa, or in Asia, they're just really important economically. So I might skip this a bit, but usually I'll talk a bit about how important it is. Sweet potato is the sixth most important crop for the human diet. So we were investing a lot in studies of its wild relatives in terms of finding genetic diversity, alternatives to sweet potatoes. So we want to find species that also have edible roots, but can be cultivated locally, for example, in some African countries, as an alternative to sweet potato. This already has been implemented in some programs of FAO, for example, in Malawi, and there are quite a few programs investing in these crop wild relatives. One of the reasons being not just genetic diversity, but also what we're talking about, the carbon footprint. So instead of having massive cultivations of sweet potato and exporting them everywhere, it would be ideal if people could just cultivate their local species, which could be an alternative. So we got lots of studies going on around these crop wild relatives, and they're really important for the challenges that we have ahead in climate change and food security. So I'll just introduce this here, because I know some people might know Lauren. So our colleague Lauren Isserman from Atlanta, she has really cool work on evolution of storage roots in Convolvulaceae, understanding how they're formed, how that affects the content in starch, in water, and so that's been really cool. It's part of this larger interest that we have in understanding storage roots in the crop wild relatives, not just in the sweet potato. Anyway, just to leave you a hint, mixing things I said. There you go. Anyway, so another thing that Convolvulaceae have in common with Solanaceae is the high amount of secondary metabolites across the family. So lots of species have some type of secondary metabolite that will have some medicinal property. So there are a wide range of medicinal uses, antipyretic, laxative. They act on the central nervous system, and if you look for them, if you search on Google, you'll find so many papers about this. Also, some species are actually toxic, so they're important to be studied for their effect on cattle. Sometimes humans eat them accidentally as well and have all sorts of problems, usually some sort of palsy of the muscles. So they have all these importance as well, phytochemical importance. And one of the things that's really interesting about them, so you will have known about this association probably of the fungi claviceps with grasses that causes ergotism, dementia, hallucination. So this has been known to occur in different times in history, where people will eat contaminated bread and it will lead to these hallucinations and craziness, as people would call it. This is quite well documented in grasses, but we're finding that this happens as well in Convolvulaceae, but with a different association, right? So 42 species of this family have been reported to have an association with the fungus periglandula, which is closely related to these claviceps. And it associates in the leaves or in the seeds, in different parts of the plant, and it gets the plants to produce in the seeds of the plant, these LSA, ergolines. And this LSA is, when orally ingested, produces very similar effects to LSD. So I don't mean to give anyone ideas, right? This is really dangerous. We know, so it's very well known as a cheap alternative to LSD because you just buy the seeds, right? Yes, I can see some people making faces. No, please don't try this at home because the results are very, well, why? Because you never know how much LSA will be in a particular seed. So dosage is really uncertain and unpredictable, and many accidents happen. So no, I'm not suggesting anything. I'm just saying that that it is known that this occurs, right? What am I doing? I just realized. No, but look, this has been known for a while. So these seeds have been used in Central American tribes in shamanic rituals. So the consumption of these seeds, this LSA, was widely used since, at least we think, since the Aztecs, for example. And when the Spaniards arrived, they tried to take it back to Europe, but it wasn't very popular. So it has to do with some, it's cultural as well. So how do you perceive this effect of drugs as something positive or negative? So for the Central American tribes, it was something religious. But when the seeds were taken to Europe, it was seen as something bad, as something negative, that you were crazy, that you were not in good health, that you were taking drugs. So it was not well accepted, and it never really took on. But it was later in the 1940s, and a famous ethnobotanist, when he visited Central American tribes, he found out about these seeds, and reportedly, I could not find confirmation, but reportedly, he was offered them to Albert Hofmann, who was making experiments for the US government on developing drugs for the military, and that he would have synthesized LSD based on a few plants, one of them being this Ipomea oil turbina that Richard had brought to him from Central America. Whether this, I'll tell you, whether this is absolutely correct, I cannot be absolutely sure. I've seen several reports of this. I tried to chase several people up to make sure that this is correct, but there are several reports, so it could be true that there is this link. Anyway, it's a very interesting story that the LSA present in the seeds of Convolvulaceae might be at the origin of the creation and the synthesization of LSD. Okay, so you want to take a deep breath? That was a lot to take in. So some cool stories about the family. So, and I would like to tell you a bit about myself. And this is the family I've been specializing in for the past almost 15 years of my life. As you can see why, because it's a really cool family with lots of things to study, lots of things to look at. And I've specialized in a particular group within the family, but like I said, I won't talk too much about that today, just a little bit so you can understand the story of my career, but I won't get into too technical, right? So, as I said, I'm originally Portuguese, so that's where it is, that orange spot. In case some people are wondering, that's okay. A lot of people ask me, even in Europe, they ask, oh, sorry, where is that? So there you go, there's where it is. It's about the same latitude as New York, but it's Southern Europe. So it's very funny for Europeans. Portugal is Southern Europe. It's kind of the hot place, very warm, almost equator for them of the rest of Europe. So anyway, that's where Portugal is. This is where I started my journey. I took biology at the University of Coimbra. And it's a small town, but it's not only really beautiful, but it's also very rich in history. And the Botanic Garden is really a central piece of that city. It's the oldest it's the oldest botanic garden in the country. It's the oldest university. It's one of the oldest libraries as well. So that really inspired me. And I have to say that was key in making me a taxonomist, right? So the way I got connected to botany and to all these very ancient history, and that shaped who I am today and the path that I've taken. So after studying biology in Coimbra, I took a course in Madagascar because I was already really passionate about plants, but I wanted more. So I found there these courses by the Tropical Biology Association. They do tropical field courses in several tropical countries. So that at MSc level, and they have it in a range of countries in Africa and Southeast Asia. They give you credits that you can use at university if it's European universities. I'm not sure if in the US that would work, but they're quite broad in the origin of the participant. Sorry, my English is breaking down because I'm getting my brain's getting tired. You have to bear with me. But it's quite a diverse. There's a lot of diversity. They make sure there's a lot of diversity in each course. There are no more than 10, 15 participants, and they just dump you somewhere in the middle of the forest for 30 days with no connection with the world, with teachers from the US, from Canada, from Europe, from whatever, just teaching you anything from zoology, ornithology, botany, ecology, conservation for 1 month. So if you're interested, I would really encourage. They haven't paid me to promote them, but that was such a good thing for me. That really was so important to me. I applied for a grant. I got sponsorship, so I didn't have to pay that much for the course. And I did a course in Madagascar. So I applied to the one in Madagascar and I got sponsorship with their help. And I really encourage anyone who has an interest in experimenting with tropical biology to do something like this. Okay. They should pay me for this, but no. I'm just too passionate about it. I just think everyone in this life should do it, even just for fun. Okay, so I did a master's degree in ecology, although my thesis was already in taxonomy. So that was my first contact with Convolvulaceae. My supervisors had a project on Convolvulaceae from East Timor, and East Timor is in this little island near Australia, just north of Australia. I didn't get to go there, but I worked with the plants they had collected there. And I did a small work on the Convolvulaceae from that place. And that's my first contact with Convolvulaceae and with proper taxonomy, actually working with taxonomy. And we discovered actually a new species among the herbarium material, which I dedicated to one of my mentors in Portugal. It's a prominent African botanist. But anyway, that was the highlight of my master's degree, that we got to discover a new species from that island. So after my master's degree, I wanted to continue to work on Convolvulaceae, and that's how I ended up in London. So I contacted a few researchers who worked on the family, and I said, I'd like to do a PhD. Would you help me write a grant? So I got in touch with someone in London at the Natural History Museum and someone in Singapore Botanic Gardens, Dr. George Staples, and they helped me put together a grant proposal, and I got funding from the Portuguese government. And that's it. I just left. I just took off to London to do this PhD in plant systematics. And there were several institutions involved in this project. So I had to do a lot of the project management myself. I had to manage all the different supervisors. I had to communicate with them. I had a blog every week where I would write everything I'd done in that week, because there was no other way that I could communicate with all the supervisors at the same time. So the blog is what worked best. But anyway, I had to develop some project management skills other than the botany skills, the systematic skills, but it was quite fun. There you go. So that was the institution that were involved. And I got to do field work because the group I was working with was pantropical in distribution. So I had to do field work in Asia. I got to do a bit of field work in Ethiopia, and only in South America is the only place where I couldn't do any field work, but just logistically, there was no more time left and I couldn't do it. So I did some phylogenetic analysis and some pollen and seed morphology, and that just led to a large taxonomic revision of the group. The group I was working with. And it was basically a project where we took an integrative approach. So we used morphological data from herbarium specimens from living collections from the fieldwork in the tropics, micromorphology, some seeds and pollen, geographic distribution, and molecular data. And we combined everything into a big revised classification of that group of plants that I was working with, and also started looking at some evolutionary studies, so morphological evolution and biogeography. The thing is, I never completely finished because there was so much to do. So the things that I work on now still kind of branch out from that project. So this is why I'm showing you this slide, because I've put together so much data during the PhD that I still got a lot going on, and I still have lots of PhD and master's students working with me on aspects, that branch out from my PhD on kind of little groups from within the major group, because so much happens. So that was kind of my starting point for the project, this thing called Meremia, which occurred all over the tropics, but it was so diverse, no one could make sense of it. They had hardly any morphological characters that were shared other than being yellow or white flowers, and these biglobose stigmas, and the pollen not having spines. So that was the only thing that was putting these things together. It was a really big mess. So what I did a molecular phylogenetic analysis for the group. We really expanded the sampling compared to previous studies. We increased the number of markers, and it basically showed us that these Meremia scintillato, we divided in up 8, well, now we're considering 10 genera, actually. Sorry, I didn't update that, but we're not considering 10 genera from there. But basically that group was not monophyletic. Meremia was not monophyletic. It was just all over the place. So let me see, I don't want to get into too many details on this, but basically we still need more genes. We're still working on that, because the little crosses is basically saying we didn't get enough support in the deeper nodes. So we do need to tap into phylogenomic stuff now to see if we can get a bit more support at the nodes. But anyway, for systematic purpose, it gave us enough guidelines to know how to break up the group, and with support from morphology, we kind of found our way. But of course, moving forward, we really want to improve that resolution. So yeah, we've divided it in a few genera. I don't need to get into detail, but there was one endemic from Brazil, and there's another one from Asia. This is the one that actually kept the name Meremia. This 10 species, a small Southeast Asian genus. Everything else was pretty much renamed. There's this other one, the Calobanthus, and this one is also from Southeast Asia. And so what happened after my PhD, I started taking up these bits, and that's how I ended up doing this postdoctoral project. So after we've split up the group into these other genera, one of the genera, so the Calobanthus, I took as topic from a postdoctoral project in Singapore. So I just left London and went to Singapore for a postdoctoral project to focus on one of these clades. And this was with a H.M. Burkle Fellowship from the Singapore Botanic Gardens. I did field work in Indonesia. I visited the largest herbarium in Southeast Asia, Bogor, and I participated at a Flora Milesiana symposium. And that's what Singapore Botanic Gardens look like, just to inspire you a bit. It's absolutely gorgeous place. Their orchid collection is absolutely fascinating. I don't know if they're the largest. They say they're the largest. I don't know that they are, but it's pretty impressive. So if anyone likes orchids, it's a must go. It's fascinating. It's a very beautiful place. So this is where I did my postdoc, so I could focus on that Southeast Asian genus. And so I worked in the herbarium. This is what the herbarium looks like there. It's got quite a big collection, but not as big as the Indonesian one, okay? So if you do want to do something in Southeast Asia, the one in Indonesia is probably the best option, but Singapore it's pretty good. It's good help. and it's very pretty. Anyway, so that's what I worked on for my postdoc in Singapore, the Southeast Asian genus with this distribution. That's what I'm working on right now. So I've got a, oh, did I hit that? Oh, no, there you go. So I now have a PhD student in Thailand who is following up on my PhD studies and focusing on this 3 genera. So he's going to finalize the things I started from my postdoc. He's extended the sampling because he collected in Vietnam and in Laos and these other countries. So that's how that's going in that direction. So that's sorted, right? The Asian stuff sorted. I put in someone else's hands. I done. So in 2015, after that postdoc, I moved to Brazil to work. Well, I got a postdoc grant to work on one of the other genera from my PhD that we had described. And that's what it looks like. As you see, it's mostly the white flowered stuff. So something that people thought were just a big group, when you start separating and breaking it down, you really see how morphologically they're consistent, you know, how they're a pretty little unit. So these South American ones, they all have these small white flowers with compound leaves. And so we described that genus in 2017. And I've just been working on that in Brazil, getting students to work on it as well, and do more field work, collect more samples, do more lab work, look at more herbarium specimens. Oh, yeah, I took off that slide, but I've got a PhD student in Brazil as well working on this, and an undergrad doing some conservation assessment for the species. Right, so after Brazil, do you need a glass of water? Should I stop? Sorry. I'll get a glass of water. Okay, so I wasn't done yet, right? I've sorted out the Asian stuff. I've sorted out the American stuff, but I hadn't worked out the African bits yet. So I had some contacts in Belgium, and I started working on the African species of this group with them. For those who don't know, Mesa Botanic Garden is a very big center for African plant collections. So that's the place to be. Other than Kew, it's the place to be if you want to work on African botany. So there I went. I just, at this time I had my little baby. I took him and said, well, let's go to Belgium. So he was that much of a baby. That was fairly recent. He was a bit grown up already, but he took him with me. And I started in Belgium what I thought would be a postdoc on the African Moremioids, as we say. So this is the Botanic Garden in Brussels, in Belgium. It's so pretty as well. It's got a proper castle, like a real castle, and fantastic glass house. Yeah, it's just an exceptional experience as well. And the herbarium, as well, as I said, it's really strong in African collections. And I've started helping them prepare the treatment for Convolvulaceae for Central Africa. So that includes Congo, Rwanda, and Burundi. I didn't do any field work, but I just worked with their collections. And I worked in collaboration with a Congolese colleague who came over for a few weeks a year to work with us on this treatment. And this is about to be published now. It's almost ready, but it's quite comprehensive. 23 genera and 145 species. That was really cool work. And so, as I said, I was getting settled in Belgium, and I was about to start a postdoc. Oh, yeah, we also described the new species from Congo. If you're interested in that. Yeah, so there I was, thinking I was going to stay for a while. I was going to do a postdoc on phylogenomics of Convolvulaceae at the University of Ghent. We're going to look as well at the evolution of storage routes with a collaboration with Lauren Iserman from Atlanta Botanic Gardens. And we got everything sorted. But then I got a job at Kew. So that never happened. That never happened. So I just jumped on that opportunity. I took the job at Kew as taxonomist in the Africa identification and naming team, which was perfect for me because that's what I was heading, working on African flora. And it's been something I've been wanting to work on since my degree, since my graduation, since I was in my hometown. So that was absolutely perfect for me. So for those who don't know Kew, well, I put some pretty words about the institution, but it is really the place where you can do anything with plants. It's the institution that hopes to inspire the world to, as they say, to unlock the potential of plants. And we work on describing the biodiversity, but also conserving the biodiversity. And more recently, there's also an investment in exploring the uses of plants, so but something's called the natural capital. So we have a bit of everything. It's really exploring the potential of plants in every sense, always thinking of the challenges of that particular period in time. So Kew has always had in history this role of looking at the challenges of its time and trying to address them. So in this case, we're worrying about food security. We're worried about tackling the climatic emergency. So again, Kew, we've been refocusing our scientific strategy towards those challenges. Okay, so Kew, actually, most people will know the garden from London, but actually Kew has 3 centers. So one in London, and then we have another center a bit outside of London in Sussex, which is the Millenium Seed Bank. And Kew also has a permanent research station in Madagascar. So these are the gardens in London, which I think most people have seen or heard of, or if not, yeah, definitely you should. It's huge garden, lots of exhibitions, quite a few glass houses, tropical and temperate and palms and everything you can think of. You can spend a week in there if you get lost. You'll just sleep there, wake up and still have stuff to see. It's absolutely inspiring. And that's why it's kind of a world reference in what it does. It's highly popular in Europe as a botanic garden. People come from all over just to visit as a park, you know, as an attraction, not just for the collections. And then the other center I told you about, Wakehurst, is the Millenium Seed Bank. It's usually referred to as a partnership because it's a collaborative infrastructure. So we've got 2.3 billion seeds, but actually these come from the partnerships with 95 different countries at the moment, and it's basically an international effort to conserve as many species as possible in the world. And the advantage of that is you can conserve a lot of genetic diversity through the seeds at a reasonably low cost. Now, we're also kind of refocusing on crop wild relatives and making sure we conserve as many seeds as possible of relatives of important crops, of these native species. So again, sweet potato and Convolvulaceae play an important role. And we're trying to collect as many native species that are closely related to sweet potato as possible to integrate in this seed bank. And then the other center we have is the KMCC, the Kew Madagascar Conservation Center. It's the only Kew research center outside of the UK with the main office in the capital, but quite a few local teams in other cities. And they're growing. They've been growing a lot recently with lots of different projects, mostly conservation, but also taxonomy. So, okay, and then we have the herbarium, which is located within the Kew Garden. So it is based in London. And we have 7 million specimens. It's supposedly the second largest herbarium. There's a bit of a dispute with Paris because depending on how you count the specimens in Paris, Paris may have 9 million or 4 million. So of course, Paris maximized and they say, we have 9 million. We definitely have 9 million. It's definitely more than Q. And then Q goes, no, 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 no. You're not counting it right because you're counting this other collection that you shouldn't count. You really only have 4 million. So there's a dispute. So that's why I didn't write it is the biggest or the largest, because I don't want to offend anyone in Paris. But we definitely have a really big herbarium, huge collection. It is a reference for tropical botany. So most of the collections are from the tropics, and Africa is definitely the largest of the collections, although we do have quite a significant part from Asia as well. Right. So for those interested, this is also the link to the herbarium catalog. This is how we work. And the herbarium has been growing. So it started with just one wing in 1877, and then up to 2009, we've been adding different wings, and we're just taking over the whole building until other people who work there can't stand us anymore. Just taking over bits and bits of the building. And we have now 5 wings. So what does our department do? So we have a particular department within the Science Directorate, which is the Department of Identification and Naming. And we do, so, and then within the identification and naming, we have the Asia team, the Americas team, and the Africa team. And what we do, all the 3 teams, is taxonomic research. We do collecting expeditions in the tropics. So we particularly work in Africa. A lot of conservation, we do specimen identification. And of course, I mean, I didn't write it down, but we do a lot of partnerships as well. So we collaborate with universities, with local museums. We co-supervise a lot of graduate students. So we have a really close relationship with partners, with local partners too. We do try to keep that. And then we, in our team, in the Africa team, one of our experts actually developed this tropical important plant areas concept. If you haven't heard of it, you might just Google it. It's just this idea where you can use a certain number of criteria to help you identify critical sites for preserving plant diversity in the tropics. So what we're constantly doing is taking up data, distributional data or threat, conservation assessments of the plants, and with a certain analysis, you will identify sites that will be important for conservation. So in Mozambique, there are quite a few now, and we're tackling other countries. So there are quite a few countries already in Africa that we're tackling towards identifying these TIPAS and taking that forward into conservation action and protecting those areas. But without that data, that can't be done. So this is kind of a fast track to conservation. Okay, so yeah, just a bit of what we do. So basically, in terms of identification and naming, the specimens will arrive at Kew from expeditions, either our own or from colleagues. We have team meetings to identify the plants. We discuss, we look at floras. And then the specimens are identified, mounted, sorted, and incorporated into the collections. And they have a crucial role as research tool. The geographic distribution data is used, the character data, and it's also important as vouchers for the samples. We have a lot of projects as well within the institutions and with partners in other institutions. For example, of the Tree of Life project. And so these specimens are important as well as vouchers. You know all this probably, but anyway, okay, so back to me. So this is what I've told you a bit about me, a bit what I've been up to, about the institutions where I've worked. It's a bit superficial. If someone wants to know a bit more about any of these, just get in touch. You can follow me on Twitter, send me an email. I'm quite happy to give more information or to put you in contact with people. Quite happy to do that. We also have this, so this Twitter handle you see here, Convolvulaceae Network. We have this group of people around the world that work on Convolvulaceae. What what we have identified was that there were lots of people in different institutions around the world working with these plants, but they didn't talk to each other. And sometimes they would overlap, or they would need help, need support. And then we found out that through meeting every Friday, people could talk to each other and help each other, and students find supervisors, or senior researchers find collaborators for projects, and people apply to grants together. And we've actually just written a paper together in Taxon. So it's become quite an important part of the research in Convolvulaceae, and everyone is welcome to join us if you want to see how it's like, or if you want to know more about the family. Or you can follow us on Twitter, as I said, Convolv Network, if you want to find out what we're up to. Okay. Oh, and that there's the email as well, if you'd like to email us. Sorry. And if you want to find out more about what we do at Q or in the Africa team, you can also follow us on Twitter. There's a Q Science and Q Africa that you can follow. And I think that's it. Thank you very much. And if you have any questions, I'll be happy to answer. I hope that was okay for you guys and it wasn't too long.
Yeah, let's give her some applause, even if we just have to push some buttons, it'll be all right. That was fantastic.
I hope that was okay. It was a lot to cover. We're never quite sure, what people are interested in hearing about. So I try to talk a bit about everything. How do I get this back to non-screen sharing?
I think at the bottom you have to say stop sharing. But
I lost that bottom part. That's the problem. Where did it go? Sorry, well, you can go ahead. If you have any questions, I'm happy to answer or to chat a bit while I find out how to not share my screen.
So I'd like to ask a question. I'm Howie Neufeld in the department here, plant ecologist. Since your work is highly dependent on travel and getting specimens and so on, what is Q doing regarding the situation with COVID and trying to keep your productivity going? And what do they envision for the future?
Well, it is quite complicated. Access to the collections is limited, so only a few people can access the collections, and only in a few days a week. But of course, working in a herbarium is not too risky because people tend to work in quite isolated areas. So in terms COVID-wise, working in the collection is not the worst that could happen. We can sort of manage people coming in on different days a week or different times, and that's okay. The field work has been compromised. Conferences as well, I think, as for almost anyone these days. So we've been trying to press on with other things. So lab work continues. In terms of the taxonomy, what I've been doing personally, and a few of my colleagues as well, is to write up the data that we have. So we have already like these taxonomic accounts that we have prepared. So whatever we can, just to wrap up, where just the writing that is necessary or some analysis, we keep that going, and that's what we've been working on. Because otherwise, yeah, it's been quite slow. Access to the collections is very, very limited, and that's slowed down species discoveries and a few of the biodiversity studies we've been doing. Yeah. Unfortunately, that's the reality. Yeah. What to do? Yes. Thank you, but thank you for your question. Nice to meet you.
any more questions from anybody?
Yeah, I've got another question if no one else is going to ask. So in the Convolvulaceae, which is very fast growing, I don't know if all the ones you've seen are this way, but generally a very fast growing species. A lot of them are vines and so on. So does this group have a high or a low proportion of species that are endangered?
Mm-hmm.
Or does it tend to be pretty ubiquitous so that it's at less risk than, say, other groups?
No, we do. Well, that's actually quite interesting. What we find out is that very few of these species have even been assessed. So we even know very little about that. There are quite a lot that are threatened. We can't put our finger on what level of threat it is, so what is the exact conservation status. But for example, one of the genus, so Ipomea, which was what you will have in most of the states, it has 900 species. 900 species. Of these, only 36 have been assessed. Sorry, Rodrigo. Right, sorry. So yeah, so out of this 900, only 36 have been assessed. So that's really serious. That's really serious that we don't even know. But in terms of the habits, it also is a problem because, of course, some of these they grow very easily. They're quite widespread. They're very hard that they'll be under some sort of threat. They actually thrive on disturbed habitats. But there are so many. I would say, I can't say proportionally because we don't have the data, but from what I know of the diversity, probably more than half of the species are not like that. They have much narrower distributions. They have other sorts of habits, so they're not necessarily twiners. And yes? Filho, tens de esperar bocadinho. Tens de esperar bocadinho.
Posso estar no microfone?
Yeah, so one of the problems, actually, that's interesting, this misconception, this is erroneous idea that they don't need to be conserved, that don't need any protection, when we actually know that many of the species might. We have that impression, that idea. One of the things actually we've done recently in this paper in Taxon is we proposed to change the type species in Ipomea, which is kind of maybe complicated to explain here, but basically what we want to do is that species that are not closely related to sweet potato, that they can keep another name and that they are not called Ipomea. Because once a species is named Ipomea, it's labeled as a weed. And then you can't really convince people that they need conservation action because they're called Ipomea, so they must be a weed, they must be invasive. So the proposal we've put together in Taxon is so that we can restrict the name Ipomea to morning glories, to sweet potato, and to many of these actual weeds, which, and interestingly, and very fortunately, they're evolutionarily really closely related. So that's fine. They're a group in itself. And then we can name the other species something else. And that really helps, because if we want to convince governments in Thailand or in Uganda or in Ethiopia that these species need conservation, it really helps if they're called something else, that they're not called Ipomea. Because of this misconception that they're all weedy and they all grow really fast, so why do we need to conserve them? Yeah, so that actually relates to that paper we've very recently published just a couple of weeks ago, to go in that sense.
Anna, I'm Zach Murrow, allegedly a taxonomist, and I'm curious about Cuscuta. I've always thought that's a problematic group, but are there other parasitic groups within the Convolvulaceae? Is that the only parasitic group?
It is the only parasitic group. Yes.
Have you done any work with that group?
Not in depth. I'm currently supervising a student in Brazil who has an interest, and she's collaborating with a group in Canada who are world specialists in Cuscuta. So Mihai Costea and Sasha Stefanovic in Toronto, and they're like the specialists on that. And so I collaborate with them, but I don't know too much in detail. I know about their work, and they help me a lot co-supervise these students. What I can tell you about Cuscuta, it's really diverse. They are the only group that's parasitic within Convolvulaceae, no doubt. So for some reason, this group just diversified, and we do not find any case of convergent evolution. Some other group also acquiring that life form. Some people have defended it should be a separate family in itself. You know, they're so different from everything else. But the genomic data, well, the phylogenetic analysis of these colleagues in Canada has shown that Cuscuta are well nested within Convolvulaceae. So, I mean, even them who work on Cuscuta, they accept that this is the solution, that this is the best classification approach, that they are within Convolvulaceae. So they also have the fused corollas like the other Convolvulaceae, but they're just very tiny. Morphologically, they're very similar, but it's just that they're minute. And the life form is different, so they don't have leaves, and they're just they have a parasitic life form. There are 200 species of them, and they have so much diversity. For example, in pollen, within Cuscuta, you have all the pollen types that you find in the rest of the family Convolvulaceae. So if you have 2,000 species in Convolvulaceae, and within that group alone, you have all the variation you find in the rest of the family, that's quite extraordinary. So for some reason, it's a really diverse group. They kind of look alike for, you know, for someone who looks from afar, because they're very, very small. But what, for people who study them in detail and have described them very accurately and in micromorphologically, you will be fascinated at that. The diversity is really incredible.
Is there agreement within the Convolvulaceae researchers that those roots or the haustoria are derived from roots, or is there debate about that?
I wouldn't know, actually. For that, it'd be better to ask my colleagues. I wouldn't know. I mean, we don't have that discussion within the non-Cuscuta specialists, and I'm not too sure on what my colleagues would stand on that, to be honest. Yeah, sorry.
That's okay. Thank you.
Yeah.
Trying to follow up on that, I wonder, a friend of mine, Claude DePamphilis, who's at Penn State, studies the non-photosynthetic parasitic plants. And one of the trends is for these plants to have a very, very reduced chloroplastic genome, because it's use it or lose it. So I'm wondering if they're not doing photosynthesis and they've lost a lot of those genes, I wonder if that just means there's less burden on that side, and they're free just to have the nuclear genes change, and it allows it to be much more diverse.
Yeah,
it's an idea.
It's an idea. Yeah, that could be.
I've got one more question for you. In terms of these alkaloids within the Convolvulaceae, and are they generated by the plant or by the endophytic fungus, or how well has that been studied?
So we have this colleague Keith Clay in Tulane who's working on this, and that's actually a very good question, because that's what he's trying to find out. How much is actually from the plant and how much actually comes from the fungus? It seems like it is produced by the plants, but because different species are infected at in different parts. So some of them would be infected on the leaves, others would be infected directly just on the seeds or on the ovary. It's not as easy to find out where exactly in the plant they're being produced, to the point where then they are taken to the seeds. So I think that's the kind of the point they're trying to get at, is at what point is that alkaloid produced, and can we say exactly by whom? So I think that's a really key point that's been trying to be established.
Great.
Where and by whom?
Yeah, thank you.
Yeah.
Well, folks, if there aren't any further questions, I think we need to let Dr. Smosh take off for a second. She's got a young man who's been demanding her attention. We cannot thank you enough, and I really appreciate you taking the time. It was a lovely seminar.
Yeah, thank you. Thank you. Thank you so much for the opportunity. It's great to meet all of you. It's a very diverse group of people, different stages. Thank you so much. It's been lots of fun. Thank you. If I can help with anything, just reach out. I'll be happy to talk more to you. Thank you so much.
Thank you very much.
Thank you.
Bye, everybody.
Thank you. Bye.
Thank you, Anna, for coming. We appreciate it.
Oh, thank you. It's great fun.
Bye-bye.
Bye. Oh, oh, now I found out how to stop sharing. Good.
Just had to find. That's how it goes.
Oh, and now I can see the chat and everything. Oh, I'm so sorry.
Oh, everything was great. We really enjoyed it. I really
Good.
appreciate you taking the time. I know it's a big deal with you being so late at night. And
Yeah. What? Okay, hold on. Can you see me though? Because I can't see myself, so that's why I was wondering if you could see me.
I
But
can't.
okay.
If you
Okay.